Hello everyone. Already in part 2 we have discussed xylem, that is complex tissue. This is part 3. In this we have to discuss the other type of complex tissue, that is phloem. Then we have to see that is tissue system, epidermal tissue system, detail of this, and then we see too that is detail of ground tissue system. This is part 3, chapter 6, anatomy of flowering plant. This is part 3, anatomy of flowering plant. Let us concentrate on xylem. Already you have discussed. Now we have to see complex permanent tissue, that is phloem, also known as bast. You know, phloem, it is responsible to conduct organic food in plant body. It consists of 4 that is type of cell: sieve tube elements or sieve cell. That is, sieve cells are found in case of non-flowering plant. That is, these are the living element. Companion cells and albuminous cells. Albuminous cells, these are found in case of non-flowering one. These are found in flowering plant. Phloem parenchyma and phloem fiber. Out of these 4, the 3 these are the living one, and the 4th one, that is non-living one. Is it right? Remember, sieve tube, companion cell, these are found in angiosperm, and these 2, that is sieve tube is replaced by sieve cells, companion cells replaced by albuminous cells. These are found in case of non-flowering plant, that is pteridophyte, and in case of that is gymnosperm. Is it right? In case of that is gymnosperms. Okay, come to the next now. That is, here we have to say one by one. First we have to say sieve tube element. This again elongated tubular conducting channels of a phloem. These are formed by fusion of sieve cells. I can give a rough diagram here that this is one sieve cell. Here it is having a thin layer of that is cytoplasm. Peripheral cytoplasm is there, but here in this case the nucleus is absent. Remember, nucleus is absent. This is the cytoplasm, thin layer of cytoplasm, and these cells will be placed one above the other. This cross section is known as, that is having sieve plate here, sieve plate, right? And in this we have the small, that is pores, known as sieve pores. So we have to see detail. Remember, it is formed by fusion of sieve cells, the tubular structure, and these are arranged longitudinally, and these are associated with companion cells. Here attached to this one is the companion cells. These are the companion cells, which are the living one, having large nucleus, and these companion cells, they have control over the sieve cells element. Come on, the end walls perforated sieve-like. This one is, as I told you, mature sieve tube. The nucleus is absent, but peripheral cytoplasm is there. As I told you, that this is the thin layer of the cytoplasm we have, which is towards the periphery, and otherwise it is having that is, here we have, that is, this is a large lumen we have. This is the large lumen, because through lumen only the organic substances will be transported. Central vacuole is absent. It is function of the sieve tube is controlled by nucleus of the companion cells, as I told you. P protein is important one. P protein are seen evenly distributed throughout the lumen of sieve tube, and it is responsible, that is healing of wound, and it is responsible for, that is movement of material. Important P protein. At maturity, what happens? That these pores, that is, they are impregnated with callose, a carbohydrate, which may block the sieve tube, that is during winter season, and when the seasons will be favorable, this will be dissolved, and in that way, that is thorough, that is passage will be formed. Sieve elements are advanced type of conducting vessels, conducting tissue found in phloem. These are present only in angiosperm. I told you, in case of pteridophytes and gymnosperm, these are being substituted by, that is sieve cells, right? Come to the next now. That is the same thing we have here. You see here that this one is the sieve pore. This is the sieve pore we have here, and these are the companion cells, and this structure, that one is that is sieve tube 1. This is one tube, this is another tube, and these are the companion cells, and this one is the phloem parenchyma. I can make one more, that this is the phloem fiber here. In between the phloem fiber, that is, these are the phloem, that is parenchymatous cells. So this one is parenchyma, that this one is companion cells, and this one is the sieve cells. Is it right? Come to the next now. Companion cells. Come on. Companion cells, what do you mean by? I told you right now. That is companion cells. Let this is the sieve tube we have here. Repetition again. That is, this is the sieve tube. These are the sieve cells. And attached to this, these are the companion cells which have, that is, large nucleus. And these are associated with sieve cells. That is lengthwise, longitudinally. They have dense cytoplasm. And they will control the, that is, function of the sieve tube. Specialized parenchymatous cells closely associated with, what is this? This one is sieve tube. This one is, that is, companion cell. Right? Dense cytoplasm, prominent nucleus, and the nucleus that controls, that is, functions of metabolic activity of, that is, sieve tube. Companion cells, they help in maintaining that is pressure gradient of the sieve tube, and because of this the sieve tube will function. Important one I told you in the beginning also, companion cells are absent in pteridophyte and gymnosperm, and these are being substituted. Can you recall? These are being substituted by, that is, we have here albuminous cells we have in case of pteridophyte and gymnosperm. Important this one is, both sieve tube and companion cells, these are known as sister cells because they originate from the same parental cell. That's why we can say these are related ontogenetically. That is, these are sister cells because they develop from the same mother cells and therefore known as sister cells. Okay. Come to the third living tissue. This is also the living one. This is also the living one. The third one is phloem parenchyma. Parenchymatous cells these are. Cylindrical these are. The cell wall is composed as usual of cellulose and lignin is absent here. And the function of this is to store the food material, other substances such as resin, latex and mucilage. And important again here is, that is, phloem parenchyma is absent in most of the monocot and some herbaceous dicot plants. This question. Okay. Come to the next now. Phloem fiber. As usual, I told you in the beginning also in sclerenchymatous fiber, phloem fiber, that is long, elongated, that is pointed structure is there. Thick cell wall is there, made of lignified, and the lumen is less. Elongated, unbranched needle-like structure. Commercial fiber, jute, flax, hemp, and these are phloem 1. And that they provide mechanical support to the, because other tissue are non-living one, they do not have lignified cell wall, so mechanical support to the phloem will be provided by, that is, phloem fiber. Primary phloem, as usual, it is present since beginning. Since beginning means that this is the vascular bundle here, this is the phloem which is present since beginning, and here this one is the xylem, that is primary xylem, primary phloem, and secondary phloem will be formed at the time of, that is, secondary growth. Is it? That phloem we can divide in the same way, protophloem and metaphloem, depending upon, that is, diameter. Yes, diameter of, that is, sieve tube. Is it? You know, as I told you in xylem also, diameter if is less, protophloem, and if it is of larger diameter will be called as metaphloem. Okay. Can you tell me now, that is, mechanical strength is because of xylem or because of phloem? Yes, it is because of xylem, because in xylem we have 1, 2 and 3 tissue. These are non-living one. These are having lignified wall, while in phloem we have only 1, 2 and 3. These are living, and only what we have, that is, only we have, that is, fourth one, that is fiber is, that is, lignified one. Okay. Come to the next now. Now we have to enter to the new topic, that is tissue system. That is important topic of this part. That is different tissues, yes, associate to work as a unit. It is called tissue system. You have seen that is tissue, then you have seen that is complex tissue, and now when the different tissues, they will be put together to perform one function, that will be known as tissue system. So there are 3, that is, tissue system found, known as epidermal tissue system, ground tissue system, and that is vascular tissue system. Come on. That is, we have here, let a transverse section is being cut. This is the outermost, which is known as epidermal tissue system. In the central mass of this, which include that is vascular tissue system made up of xylem and phloem vascular tissue system, and in between these two we have the tissue excluding epidermal tissue system and vascular tissue system, that is known as ground tissue system. Okay. Now we have to go one by one. We have to see now, that is, first we have to see that is epidermal tissue system. Epidermal tissue system, you see now. Come on, as I told you, it consists of epidermis, because it is the epidermal tissue system. This is the outermost one, protective one. So it will have epidermis, and we have that is stomata, and we have the epidermal outgrowth. We have to discuss three things here: epidermal tissue system, that is epidermis, stomata, and the epidermal outgrowth. Epidermis, outermost layer of plant body made up of elongated, compactly arranged cells which make continuous layer. Let I am making here that this is the epidermis, that is, these are the elongated cells. Continuous layer is there, and it is made up of, that is, these cells, compactly arranged continuous layer. Usually this is single layered, but in some cases it may be multi-layered also. It may be having more than one layers. Some examples are there, multi-layered. It is covered with waxy coating, that in some cases it is having a waxy coating outer side known as cuticle, especially the aerial part of the plant, like that is stem and the leaf, they have this waxy coating, cuticle, but in case of root the cuticle is absent in root and in case of hydrophytes. Is it right? In some monocot leaves, specialized epidermal cells, they have become bulliform. We have to see in detail in leaf also. But you see here in case of leaf epidermis, that this is the leaf epidermis. Some of the epidermal cells in case of leaf, they have become, that is, large one, and these are known as bulliform cells in case of leaves, especially in some monocot leaves. And these bulliform cells, they help, that is, in rolling of leaves. When the scarcity of water is there, then the leaf will be rolled up and the cells will become flaccid. In root epidermis, it's better to call root epidermis is known as 𝕃𝕀𝔻𝔼 or 𝕃𝕀ℕ𝔼 layer. Is it right? It is called 𝕃𝕀𝔻𝔼 or 𝕃𝕀ℕ𝔼 layer. Come to the next now, that is, we have to see now, that is stomata. Okay, stomata, you see now. What we have in stomata, come on. You are familiar with this. You must have prepared this slide of stomata also. What we have in stomata, these are, that is, small openings found in epidermis of green aerial parts of plant. If this is, that is, herbaceous one, then what will be there in this? That may also have stomata, and stomata usually are found in case of, that is, leaves. They regulate the process of transpiration and gaseous exchange, especially in case of, that is, leaves, and the stomata are absent, that is, in root, because there is no need of gaseous exchange there in root. The pore of each stoma is surrounded by two kidney-shaped cells, and these cells are known as guard cells. Is it right? These are called guard cells. Come on. And these guard cells, these are living and they contain chlorine. I will give the detail of this. See now. This is one guard cell, that is the other guard cells. So these guard cells, these are the living one. They have the nucleus and they have this, so many chloroplasts are there. In case of, that is, you see here in case of dicot, that is, these are kidney shaped. In case of dicot, these are kidney shaped one. And their inner wall, that is the thicker one here. This wall is thicker, while this is the thicker wall, thick one. And the outer wall is, that is, this is the thinner wall. So these cells are known as guard cells. In case of dicot, these are kidney shaped one. And the space in between the guard cells, that is known as, that is, stoma. These guard cells, these are surrounded by some cells which are little modified, and these are known as subsidiary cells. You see in case of dicot, this is the guard cell, that is here the inner layer, this one is the thicker one. Inner cell wall of the guard cell is thicker one, this one, while the outer one is thinner. These are chloroplast, and this is the place stomatal aperture, and near to these guard cells some of the epidermal cells are also modified, known as subsidiary cells. So here this is the case of, that is, this is, yes, stomata in, that is, dicot leaf. Is it? In case of monocot leaf, the structure is little different. Here you are seeing that these are the guard cells. The guard cells here, these are the guard cells. The guard cells, these are dump bell shaped like this. This is one guard cell and this is the other guard cells like this. These are, and here this wall is, that is, this wall is thicker. This wall is thicker. So these are the guard cells, and near to these guard cells, these are subsidiary cells. You are seeing that is, these are also modified. So in case of, that is, monocot, these are, that is, this is guard cells, these are, that is, kidney shaped one. These are, yes, come on, kidney shaped, and here these are, that is, dump bell shaped. These are dump is bell shaped. Is it? So that will be, that is, stomata in, that is, in case of monocot, in monocot. Is it right? So that was about stomata. Come to the next now. We have to see, that is now some structure related to stomata. In grasses, guard cells, as I told you, dumbbell shaped these are. In case of dicot leaf, these are kidney shaped one. Outer wall of the guard cells away from the stomatal aperture is thin. The inner one is thicker one. Important characteristics, repetition of the same. Inner wall of the guard cells, they have cellulosic fiber which help in opening and closing of the stomata. Guard cells are surrounded by subsidiary, or that is accessory cells, I told you just, and the guard cells, they regulate opening and closing of the stomata. Stomatal aperture, guard cells surrounding subsidiary cells together, all these are known as, that is, stomatal apparatus. In case of xerophytes, stomata are sunken. These are not on the surface. If this is surface of the leaf, then stomata, these are found in pit, and such stomata are known as sunken stomata, and it will reduce the rate of transpiration because already scarcity of water is there. Transpiration is greatly reduced. Come to the next now, that is epidermal appendages. You know, we are dealing nowadays, that is epidermal tissue system. Epidermal tissue system. In epidermis, epidermal tissue system, we have seen that this one is, that is, epidermis, a continuous layer. Now from this epidermis, some outgrowths may arise, that is known as epidermal appendages. In case of root, let this is root, you see some of the epidermal cells, they will get elongated into a unicellular structure, and this is known as, that is, root hair. You know, that is root hair. In case of root, that is the hairs, these are epidermal growth. These are always unicellular and these are short-lived, is it? And they increase, that is, surface area for water absorption. Root hairs are unicellular, tubular, exogenous, found in, that is, zone of maturation. You must be remembering, if this is the, that is, root hair, this is root cap, this is what? Root apex, zone of elongation, and this is called the root hair zone, from in which we have, that is, root hairs. Cell wall is thin and made up of spectrocellulose. Root hair are ephemeral. Ephemeral means these are short-lived, that is, new hairs will be formed, and root hairs, they help in absorption of water and minerals. Root hair. Okay. Come to stem. In case of stem, what is there? If this is the stem, these are the epidermis of the stem, then some of the epidermal hair, epidermal cells, they have, that is, outgrowth, which are multicellular. Remember, these are, that is, multicellular, and this multicellular outgrowth is known as trichome, or also known as multicellular hairs. These are epidermal hairs on stem, multicellular. They may be branched or unbranched, they may be soft or stiff. Trichome sometimes may be secretory also, that they may secrete some substances, and trichome prevents water loss due to transpiration. They provide protection and they also help in dispersal of seeds and fruits. This is regarding, that is, epidermal appendages. Is it right? We have to proceed to the next, that is, ground tissue system. I told you what is ground tissue system. You must be remembering, if a section is being cut like this, that this is the central cylinder. Central cylinder is made up of, that is vascular tissue system. This is the outermost one, that is epidermal tissue system, and in between the two, that is ground tissue system. Is it? This ground tissue system extends from epidermis to the center axis, that is excluding that is vascular tissue system and epidermal tissue system, or is ground tissue system. It is made up of cortex. Cortex is in between epidermis and the pericycle. It is further differentiated. You can see here, this is epidermis, that this is the, that is we have endodermis, and that this one is the pericycle. Remember, this one is the, that is pericycle. Is it pericycle? It lies, that is inside, that is endodermis. Remember, this one is, that is epidermis. So all tissue that is in between epidermis and pericycle. So it will have, what is this? This is the hypodermis one. Is it? That is known as, that is general cortex, and this is called the endodermis. Is it? Endodermis. So this cortex, and this is what? Pericycle. The cortex, what we mean by cortex, is having hypodermis, general cortex, and then it is having endodermis. Hypodermis, it is colon-chymatous in dicot stem and scleron-chymatous in monocot. I told you that provide, that is strength. General cortex, it is made up of parenchymatous cells. You know that we have to make the diagrams here. These are the parenchymatous cells like this, and that is, this is general cortex. Endodermis is one of the important layer. This is endodermis 1. You have to see the detail of this endodermis. In case of stem, that is, it is single layered, made up of parenchymatous, barrel shaped cells. These are like this, barrel shaped cells like this, and these are compactly arranged. In case of root, you see that they have thickening on radial and, that is, tangential wall. This is the thickening. This thickening is very important. In inner and the radial wall of the endodermal cell, they have Casparian strips, and these Casparian strips, that is, these are having this suberin wall. This will make the endodermis a watertight dam, and to check the loss of water. That's why this is airtight one, and it will check the entry of, that is, air in xylem vastus. One more important point here, the tissue present inside the endodermis, that is called, that is steel. It will have pericycle and, that is, vascular bundle. So Casparian strips, these are found in case of root. Is it right? You see, one more important point we have here. In case of root, we have to see in detail when we study root, that is in between, that is endodermal cells, the passage cells are present, and through passage cells, they do not have Casparian strips, and through passage cells, the water from cortex, that will enter into the vascular tissue. Water from cortex, that will enter. Passage cells are there. These passage cells are thin-walled one. Okay. Come to the next now. Pericycle. It lies between endodermis and the vascular tissue. Usually this is single-layered parenchymatous in root, scleron-chymatous or mixed with parenchyma in stem. Important point, the pericycle just opposite the protoxylem is the seat of origin of lateral roots. You see in case of root, that is the lateral branch that come out from inside endogenous, and the lateral root they come out from pericycle. In dicot root, you have to study in detail. Dicot root pericycle form part of cambium, and that is whole cork cambium. You have to study when we deal with, that is, secondary growth. Pith is the enormous central part in dicot stem and monocot root. This is well developed. In dicot stem, the monocot well developed one. In case of dicot root, this is less developed, and it is absent in, that is, monocot stem. This pith is absent. Pith is absent. Is it right? So this is about this part 3 of chapter 6. Thank you.